of course, there were Professor Komisarenko presented, the very complex description of medical approaches and biophysical, biotechnical approaches to the fight with diseases. But diseases can be also prevented on less complicated level, simply by using a different approach and taking lactic acid bacteria to control bacterial growth and also fungal growth. Can I see the next slide, please? Well, this is, the story starts with our PhD student from Mongolia, who was asked to look for the bacteriocins in the dairy products of her country. Bachargal, what is his name, is actually a professor of biochemistry and biotechnology at Ulaanbaatar University, State University. She brought several samples, several aliquots of different Mongol products, from which one, which we classified as Enterococcus durans A5.11, demonstrated a very powerful antibacterial activities. So we were interested to characterize it and to purify it, characterize it, and to study it further. We have extracted from the medium, usually with organic solvents, this, and then put it on the cation exchange chromatography. And here is this peak presents the activity. The next steps was the reverse phase chromatography. We collected this peak and put it on reverse phase chromatography, collecting this peak. We have after we checked the activity against the reported strain LB340, and here there was activity, found activity. Well, can I have next slide? Can I have a next slide, please? Well, of course, the slide before. Sorry. Oh yes, our investigation continued. Also, thanks to other trainees from in our laboratory, and this in this case it was a Spanish fellow who came for a postdoctoral stay. He was from Instituto de Grasas in Sevilla, and he was interested, realizing how efficient this Enterococcus durans was against the bacteria. He wanted to check it if he can use it for the control of the bacteria in the fatted sunflower meal. And he asked us to give him this strain. What we did. The results were quite surprising. Without Enterococcus durans. Sorry, what is going on here? Without Enterococcus durans, the Petri dishes were covered with the colonies of the molds. With Enterococcus durans, the size of colonies and the density diminished. Without agitation, it is here, and with agitation, it almost disappeared. The colonies almost disappeared. Can I have a next slide? Well, of course, all these molds from the sunflower meal contained many, many different species of the mold. So it was, well, we had only the information that the strain was anti-mycotic, but we didn't know nothing about the exact specificity of the strain. So we tried first on the Fusarium curvorum, and here we realized that this enterocin, which we call duransin, was active against Fusarium curvorum. Can I have a next slide? We have, as you have seen before, we have purified it and we have sequenced. Apparently, we found two different peptides of the mass around 5,000 Daltons. And so we have synthesized, one was shorter, which we called peptide 43, and the second was longer. We called it 44 because it contained 44 moieties. And we synthesized the halves of these peptides, what we called N-terminal 43 and terminal 44, C-terminal 33, and C-terminal 44. The activities of all these peptides were tested at final concentrations of 2 millimolar. Please, the next slide. And what happened that here we see that on the activities, antibacterial activities against Listeria ivanovi and mushroom activities against the mold or the yeast, the Biopolymères hanseri. The Biopolymères hanseri is a very nasty mold which is surviving dry conditions, high salt and so on, and he's very hard to get rid of from the many food products such as sausages and cheeses and so on. So we can see that here the results show that antifungal activities of N-terminal peptides, truncated N-terminal zeta and N-ter44, are superior than the peptides 43, 40, 44. So the halves of these bacteriocins are more efficient against molds than the full peptides, and what is totally reversed in the case of antibacterial activities, when the full bacteriocins are much more efficient than the short versions. Please, next session. We have looked at the structure of these peptides, and we have seen that in aqueous medium, peptide 43 has a spectrum typical of alpha helical peptides, and peptide, its half and terminal is already typical, has a structure already typical of disordered peptides. Can I have a next slide? Well, of course, there are few differences. They are almost identical, as you can see. Compare the sequences. So, with some conservative mutations, but also there are the differences of the charges and which are minute, but however important. Can I have a next slide? So to see the activity and the mechanism of activity of these two peptides, we were using blue Nile A staining, which is a fluorescent dye used in cytology, which is lipophilic. It stains specifically membranes and polar phospholipids in particular. So we have done it, with culturing the Baryomyces hansenis 72 hours and processing it, centrifuging during 10 minutes and staining it 10 minutes and dissolving in phosphate buffer, resuspending it and looking at what happened in optical and confocal observation. What is if the cell membranes are disrupted, then of course the blue Nile gets in and colors the inside of the cell. Can I have the next slide? So what do we see? This is control. This is when we use the peptide 44 on the Baryomyces, when we use N-terminal 44 and C-terminal 44. So it's evident that the full peptide doesn't work on the Baryomyces, but the halves, yes, they do. Here is this confocal microscopy, which confirm what we see in optical microscopy. Please, the next. Another approach was staining with propylium iodide, which is a fluorescent compound intercalating in nucleic acids, DNA or RNA. It is staining nuclei of cells having lost membrane integrity. So we have processed in the same way as with previous experiment and looked what happened in optical and confocal microscopy. Can I have the next slide? The picture is confirming the entry of the bacteriocins into the cells, okay? And we see that in some way also the full compound some extent gets in. But this is it. It concerns peptide 43. Then before one was peptide 44. So the peptide 43 is somewhat more active against the Baryomyces. And we have the entry into the cells and coloring of the DNA. Of course, also in this case, we can have a little bit some cells which are already disrupted. Can I have the next slide? We have looked at the influence of these peptides on the growth of the Baryomyces staining. We here this if we use the 10 to 100 micromolar solutions, there is no influence. If we increase the concentration of the tested peptides or the halves, we see that the start of inhibition at the 1 millimolar, which is, however, quite important concentration, we see that the most active is the C-terminal 44, and the peptide here, it's full versions as well. Can I have the next slide? We have looked at what happens with the Baryomyces by electronic microscopy. This is control. Then what is happening when we give the 2 millimolar concentration of peptide 44. So we start to see protrusions of the of the cytoplasm outside of the cell, and some disorganization of the interior cytoplasm. If we look at the peptide 44, it's half the the morphology of the cell is dramatically changed, and the same with the C-terminal half of the same bacteriocin. The cell simply is destroyed by the applied bacteriocin. We have rechecked or debaryomyces Hansenii and Yarovia lipolytica, which are both the molds, and can you click, because it will show the yes. Well, so there's some problem with this slide. Here we have peptide 44, half of the peptide 44, which are the halves of this N-terminal 44 and C-terminal 44 show evident antifungal activity at concentration of 2 millimolars, and the other synthesized peptides are inactive at this concentration. Can I have a next slide? Okay, so since these results were relatively interested, we were contacted by the major French dairy producers interested in the conservation of their products. These industries are Danone and Standard. And we launched the study of the fight of the applied lactic acid bacteria against several fungal infections, which are very negative for the product conservation, such as Penicillium roqueforti, Penicillium expansum, Debaryomyces hansenii, Mucor plumbeus, Geotrichum candidum, and Fusarium. And these were studied on the yogurts, and we have also looked at the influence of the secondary metabolites of the yogurts. Okay, so this was succinate. Can I have the next slide? So the acetic acid, hexanoic acid, lactic acid, succinic acid, phenylgalactic acid, pyrrolidone 5-carboxylic acid. Can we return? No, return higher. Return to one more. Yes. So what we can see, why we have looked at this, because it was found that the strains producing hexanoic acid were very efficient against these mycotic infections. So you can see, well, here we don't have too much activity. We see that the acetic acid and lactic acid are quite efficient in controlling the fungal infection, and also the most efficient are derivative of hexanoic acid. Can I have 2 slides more? Yeah, good. So we here we have looked at these continuous these studies, looking at the yogurts produced with Lactobacillus bulgaricus and classical, and which were of course infected after 6 weeks after contamination with fungi. However, when they were produced in presence of Lactobacillus carbinensis, a heavy producer of hexanoic acid, the yogurts remained untouched by the fungi. So this was quite important finding, even if it looks quite modest. The strain which we selected of Lactobacillus carbinensis is now used by the dairy industry in production and conservation of yogurts. Please, next slide. We have also looked if the matrix it has any importance in conserving activities. So if we added Lactobacillus carbinensis, there is no problem with the infections of the yogurts with Yarovia lipolytica. Can I have the next slide? We have also looked at if what happens on jellified fermented milk. So here we have non-acidified milk, control, fermented milk, well, a little bit later, with addition of lactic acid. We have quite good protection. If we give simply HCl at the pH and go to the pH 4.2, in the case of penicillium expansor, so we have quite significant protection. So the acidification with lactic acid or with HCl is also a good way to protect against infection with penicillium expansor. If we look at the Biopolymères Hansenin, we have obtained almost the same picture with the best protection by lactic acid. With Yarovia lipolytica is just less efficient. So only slight delay is seen on Yarovia lipolytica in these conditions. Can I have a next slide? So fermentation time effects. So if we have this with milk, infected with the Biopolymères Hansenin, there is a fungal infection going quite well, quite rapidly. If we add the lactic acid, it is delaying the progression of molds. In the fermented milk, it's relatively stable, and with added lactic acid, it is almost entirely protected against the Biopolymères Hansenin. So as you can see, antifungal activity is observed during all milk fermentation time with Lactobacillus carbinensis. Okay, because however, the lactic acid alone is also active against the Biopolymères Hansenin. Next slide. So The first summary is just the activities of antifungal compounds produced during growth of Lactobacillus hansenis, previously isolated from raw cow milk, which was found to possess strong antifungal activities, were tested. A pH-controlled batch fermentation in enriched milk protein medium with immobilized cells of this bacterium was carried out in the presence of disrupted cell of the Biopolymères hanseni and Chlorobacillus lactis. After 7 days of fermentation, cell-free culture supernated with robust antifungal activity against the Biopolymères hanseni, Penicillium expansum, Penicillin roqueforti, were obtained. These cell-free supernatants contain high amounts of organic acids such as lactic and acetic acid, as well as hexanoic acid, which the later revealed to be the most efficient antifungal compound. The antifungal activity of hexanoic acid was influenced by the environmental conditions, including the nature of matrix and pH. Under the condition tested, even if organic acid were key antifungal agents, they were only part of a complex mixture of various molecules that acted in synergetic manner. So the morale of this is that the strategies of lactic acid bacteria are very, very, complex. Not only the bacteriocin may be antifungal, but also whole population of secondary metabolites, often, fatty acids and compounds which are acidifying the medium. In the next, the following studies, the results we obtained were mobilized the Association of French Bakery Association to ask us to study the ways to protect the products, and what we did with positive results. Since there is some time economy important for the conference, I will stop here and not describe all these bakery things. Thank you very much for your attention.
Thank you very much, Professor, for your interesting lecture. We have a question from Andrei Medlich. Please ask.
Bonjour, Professor Thomas. Ça va? Vous allez bien?
Andrei, yeah, bonjour. It's nice to see you.
Nice to see you. Thank you very much for your brilliant talk. And I have a question, if you don't mind. What do you think? What do you think? What is the best source to isolate strains of lactic acid bacteria with antifungal activity?
Well, the best ways, as you know it from your stay in our laboratory, is look for the isolates from already traditional food products, such as dairy and such as fermented vegetables. Also, from starting fermenting grapes. Our experience shows that the success rate is about 5%. So if you collect 100 aliquots for 100 different products, you have some chances to find 5 or 4 strains of interest. Okay, so there's a lot of work to isolate them. So if you get it, you really have an interest in keeping them.
Okay, thank you.
Thank you very
actually,
much.
we are trying to continue our research work with our Enterococus, and we also found antimycotic activity, but against one pathogenic fungi, Fusarium and Alternaria. So we are
Yes.
trying to continue.
Very nice, Andrei. If you have anything more synthetic, written on this, I will be very happy to look at this.
Thanks.
Good.
Thanks a lot.
Thank you very much, and all the best. All the best to everybody.
Yeah. So we have a question from chat from Joseph Michael Ochingo-Dur. Does this find confirm the ability of yogurt to resolve oral geotrichosis trash?
It may help, yes. If there is a well fermented with a good strain, yes. But not every yogurt is fermented with Hanseatic, with strains which are producing hexanoic acid.
Thank you, Professor. And next will be Mikhailo Ivansov.
Good afternoon. I have a question. How long does it take you to complete this experiment?
Yeah. Well, which experiment? To find out the right strain and the most interesting strain, it takes years. Our Mongol trainee, very good one, came to our lab in 2005. Okay, and we have discovered about this strain about 2007 and so on, and start slowly developing it. At the beginning, it was only the antibacterial, very good, very powerful antibacterial tool. One shouldn't forget also this aspect of the problem. The humankind needs also the antibacterials, not relying only on the antibiotics, just the natural antibiotics such as those bacteriocins. So the Enterococcus durans is excellent tool to secure the safety of dairy and other products.
Thank you for the answer.
Yes.
Thank you. Any more questions to Professor? Mykola Rogalevsky next.
First of all, thank you for your lecture. And these bioactive molecules, bioactive molecules are added in the last of production line, or they are they are put in the other product state.
Yeah, that's a very good question. To start with, the addition of any chemicals to the food is very strongly regulated. So the addition straight of, for instance, hexanoic acid or whatever, it needs to be very heavily controlled. Okay, the secondary metabolites of lactic acid bacteria are produced in minute quantities, and so minute that they are very, very hard to identify in the very complex fermentation medium. That's why we were using the purified chemicals, you know, these to test how the molds are applying to them. But for the use in the real food, one needs to be extremely careful. Okay, so the manipulation of that kind are very delicate. Okay, it's much easier, much safer to ferment it with a given strain. Okay, which has a spectrum of these compounds more or less known, more or less controlled, than to, you know, add by the spoonful of the different compounds. So the it's a very, you know, delicate balance. However, I underline that is a very, you know, delicate way to improve the safety of the foods and the health of the population. Because if you are protected against food intoxications, you are much safer and much healthier. Even much more stronger protected against all the viral infections and so on. Do I answer well your question?
Yes, thank you very much.
Okay, thank you very much, Professor. We have time for one more question. Who wants to ask something? Okay, I have one question. I think it will be the final. Professor, did you discover the internship with your peptides and classical preservative agents such as nisin, or you plan to do this?
Well, nisin is already very well known. However, there are several different varieties of nisins. There's almost the family of nisins. Well, internship, it could be interesting whatsoever. You know, all progress in this domain can be very, very positive. Yeah. But finishing also, I need to underline our strong engagement into what happens around Odessa and what happens in your country and in that part of Europe. So the organization of the school is also a very positive side. I congratulate you again and wish the best, you know, ending, finalization of all the school, and wish to all the scientific, professional, and existential success and achievements to the organizers and to the attendants. All the best.
Thank you very much, Professor, for your very interesting and very practical related lecture.